Good morning, ladies and gentlemen. My name is Soo Kim, CEO of Cell. As you know, that we are a developer of CAR T immunotherapy, and maybe you have already listened to this, but it is considered a dream cancer therapy or cancer therapy of hope. And you might wonder why this is the case. And I'm going to show you and explain the principle of CAR T therapy and what we're currently developing. If you look at this image, this is the imagery of terminal cancer patient. These patients usually have already used all these options and they do not have any more options left. If you look at the images, all those cancers are dominating the body on the left image, and then within 1 month of the administration of our treatment, it's almost gone and treated with the cancer. And there are two approved CAR T options. One is C or CD19, and there are four different products: Kimli 19, and Naticartas, and then Brianzi, and then Yescarta. And then early this year, BCMA CAR T was approved as the second type CAR T in in the world, and this is called Ave CAR T as well. So using single medicine and being able to treat a cancer, this is called single therapeutics, and it is basically used as a single dose administration in order to remove almost all cancer cells in a terminal cancer patient's body. It takes about 10 years to come up with a new development. That's true. That's common. However, if you think about Yescarta's development, after pre-inhibiting to the actual approval, it only took us 6 months. Why is it possible? It's because single dose administration, and within 3 months, you can check and identify the reaction of patients' bodies. And if you think about early this year, in Korea, CAR T clinical studies were not going on at all. In China and in the US, there were more than 300 CAR T clinical trials. So Korea was excluded from the scene, but for the first time in Korea, we received the approved approval, and as a result, we're now going through the early stage of CAR T clinical trial. Now, let me go to the main principle of CAR T therapy. On the left, there is natural T cell reactor, and you have to understand how T cell is basically killing cancer cells. It is simplified, but there are many different molecules that are involved. There's MHC TCR complex that is recognized by T cell, and then once it is judging that it is a cancer cell, then it does immune attack. And this complicated mechanism has to evolve many enzymes and reactions, and CAR T is basically simplifying the process. What it does is that it puts in the order of antibodies, and then it activates tumor cell T cells by utilizing other molecules, and that's called CAR T therapy. The concept itself was developed a long time ago, which is the 1980s in Israel, and then after going through many different rounds of development, and now we call next generation CAR T when CAR T technology includes other molecules. Currently, commercially available and widely used CAR T therapy would be second generation. And the simple mechanism is that there's a single chain FB will recognize a specific antigen, and the signal that's recognized is going to activate the CD2 ejecta or co-stimulation domain in the cell, and it will be amplified. And then the sleeping CAR T cell will be awakened, and then that'll activate a T cell, and then it'll proliferate, and then as a result, will remove and eliminate cancer cells. By going through that phase and steps, cancer will be treated. And now let me discuss how cancer is fought by CAR-T. What's most important with the partnership with hospitals? What we used to do is basically pharmaceutical companies send and ship medicines, and hospitals simply use them. But CAR-T therapy is different. During the manufacturing system, the hospitals intervene and put impact. So basically, what you do is we do pre-treatment testing when we create and collect lymphocytes. So it's basically in the hospital, there is a dialysis medicine, a device. It's not exactly dialysis device. It's similar to that. So utilizing that device, we collect lymphocytes and then we send this to GMP of pharmaceutical companies, and then by utilizing that sources, we culture T cells, and utilizing different methods, we will introduce other genes, which is there could be a non-viral vector option or viral vector option. And by utilizing a massive culture method, we achieve the target dose for a specific patient. And after that, what they do is the factories freeze the manufactured CAR-T, and then the cancer patients will go through lymphodepletion. In order for the CAR-T therapy to be implanted in the human body, lymphodepletion process is very important. Patients' lymphodepletion has to be done before CAR-T to be injected. That is why CAR-T has to wait in the freezer. And once the lymphodepletion is done, then the CAR-T therapy medicine will be shipped to the hospital, will be thawed, and then put it into a patient's body. This is only for one patient. So think about costs, ingredients, and labor costs for this one single patient. That is included in the cost of the medicine. As a result, if you look at the right side of the image, the overall cost for CAR-T therapy, the cheapest would be like 500,000 USD, which is 500 million won. And if there is side effects that are appearing in the body, then maybe the maximum will be a billion won. And there are many CAR-T therapies that are approved, and it showed great results. But there are homework and hurdles that CAR-T therapy has to overcome. First would be even the patients that completely removed and eliminated cancer cells in 1 or 2 years, it can sometimes come back. So we have to think about how we're going to handle long-term survival of these patients. And secondly, if you think about lymphoma's treatment results are different. So lymphoma has a twice better result than cancer. So it depends on the types of cancer. Depending on the type of cancer, the treatment rate differs. So we have to understand how we're going to increase the treatment rate, the completion rate, depending on the types of cancer. And now we have to think about how we're going to more effectively target solid cancer rather than lipid cancer, because solid cancer CAR-T therapy has a very, very low rate of success. Especially PD-1 blockade antibody, if you put them together with CAR-T, we can increase the efficacy by a little bit. There are some reports about it. And in our company, we call a new technology called OVIS. And now let me go into detail about OVIS. So now you have to think about why CAR-T is not effective for solid cancer. There are many different explanations, and this image has some content. So what we need to focus is that there are immune cells in the tumor, and then because they interact with cancer cells for such a long time, PDR immune strengtheners representation is already heightened, and that is why CAR-T therapy is not as effective. As a result, PD-1 antibody is now jointly used with CAR-T therapy in order to overcome that challenge. And what we're trying to do is how we're going to inhibit the immune strengthener. And as you can see from the left image, we represent CAR but we also make sure that PD-1 and TIGIT to be inhibited. So theoretically speaking, CAR is right there. The CAR technology is right there, and then we also use inhibitor of PD-1 and TIGIT. So it's a combination of these two technologies. So when PD-1 and TIGIT are there, of course, a CAR will activate T cell activation. But as you can see, because PD-1 and TIGIT were already removed by our technology, CAR T can more successfully and efficiently target cancer cells. So we carried out animal clinics, and then as you can see, if you compare conventional CAR T and our OVIS-TM CAR T, depending on doses, there's high dose, medium dose, and low dose. And this is also lymphoma cancer cells. And we also use T cells of a healthy person. And for high dose, both showed great efficacy in treating cancer. But if you go down to low dose and medium dose, you can see that Ovi's team CAR T is showing better efficacy than conventional CAR T. And now next one is the pre-OC data of Ovis CAR T. We also divided them into 3 different doses. And if you compare conventional versus Ovis CAR T, and you might not be able to see the picture very well, but if you look at the 3 graphs, for the tumor version, Ovis CAR T showed a significant drop in the graph. And the blood T cell were blood cancer patients T cell. And if you go into Ovis technology, there are many different attempts and trials in overseas as well. You know, Caribou Biosciences, which is famous for receiving Nobel Prize, they are using CD19. It only specifically removes CD19 receptor inhibition. But now, instead of that, we are putting PD-1 and TIGIT neutralization at the same time. We already proved through our basic research that removing PD-1 and TIGIT at the same time can show and strengthen the results. So we received healthy blood from SNU hospital, and then we have created 100 batches already, and then we also received blood cancer patients' blood, and then we created 25 batches, and based on the results of the study, we have received the IND approval. Now moving on to the clinical trial design for Ovis CD19 CAR T, our first goal is to check safety, and then we also will check efficacy as well. It is 3+3 design. We're going to do low dose, medium dose, and then high dose. And then you can see that 3 patients that belong to cohort 1, 2, 3 is going to go through the increasement of dose administration for their bodies. And we have a very close partnership with Samsung Seoul Hospital. We will utilize GMP facilities in some Samsung Seoul Hospital. About 80 to 100 patients CAR T doses can be generated and created. Currently, we have a POC clinical trials for CD19, but we also would like to go into TPD lymphoma, AML pipelines in the near future, and eventually for solid cancer treatment, we would like to develop first-in-class CAR T therapy program. This is our mid and long-term goal. Thank you very much. But because of time constraint, would you please conclude your presentation?